Class 9 students. Subject Biology, Unit 4, Diversity in Living Organisms, Lesson 9, Economic Importance of Bacteria and Fungi. In this, as part 1, we are now going to learn economic importance of bacteria. Is it clear? Only about bacteria in this unit you will be learning. And in the next part, you will be learning economic importance of fungi. Is it clear? Now, what are bacteria? Bacteria are organisms which are too small to be seen by the naked eye. So, many of them people consider bacteria as invisible enemy because of their pathogenic nature. So, invisible enemy. Now, in the pandemic coronavirus is invisible enemy, isn't it? We cannot see, but it is still an enemy. And pathogenic means which causes diseases. That's the word here. As large number of diseases of mainly for the plants and animals are caused due to bacteria. Cultivated plants, domestic animals and in human beings are caused by the bacteria. Got it? But at the same time, it is also a fact that an equally large number of bacteria are friends of man and are useful in agriculture, industries, medicine and recycling of matter in nature. What is meant by recycling of matter in nature? So the dead becoming decay and getting again mixing into the soil. So minerals will go back to soil. That is called recycling of matter. Got it? So first we will be learning harmful effects of bacteria. So bacteria cause diseases in human, animals and plants. Many species of bacteria, they cause diseases, children, in humans, animals and plants. Got it? And they also cause suffering, pain, got it, wounds, and sometimes they also cause loss of life. They are very small, being microscopic and extremely small and very light. They enter the body very easily, either through food, water, air, sometimes personal contact when you are going by bus, touch, like that. Personal contact or through cuts, wounds on the skin. So that is how they enter into a body of organisms. So diseases in humans. For the first part, we are learning diseases, isn't it? So diseases in humans. So many diseases are there, various others. Here we are only going to mention a few. Got it? First part what I'll say is the disease name and the next part is causative organism. Got it? So typhoid fever, Salmonella typhosa, tetanus, Clostridium tetani, diphtheria, Corynebacterium, diphtheriae, tuberculosis, Mycobacterium tuberculosis, leprosy, Mycobacterium leprae, whooping cough, Bordetella pertussis. That is why whooping cough is also called as pertussis. Dysentery, Shigella, pneumonia, Diplococcus pneumoniae, cholera, Vibrio cholerae, syphilis, Treponema pallidum. Okay, and you need not learn all the spellings and the causative factors, children. Don't get worried. Oh, so many words we have to learn, ma'am, and all. It's only for your awareness. Blood infection, species of Streptococcus, food poisoning, Clostridium botulinum. Okay, these are the various diseases which we find in humans. In animals also there are so many diseases. Only a few you are going to learn in this part. Two main diseases you are learning is anthrax, bovine tuberculosis. Anthrax is mainly caused due to the bacteria called this Bacillus anthracis. Got it? And another disease you are going to learn is black leg, which is caused by Clostridium chovio. Got it? And one more you are going to learn is bovine tuberculosis. The anthrax symptoms, children. By looking at the animal, we can we can easily identify. What are the symptoms? Swelling on the body, reduced milk yield. Suddenly the milk yield will come down. Got it? In the case of tuberculosis, lungs get affected and the animal starts coughing in a husky way, dry, husky cough. Got it? These are the symptoms shown in the animals. Now we move to some of the common plant diseases caused by bacteria. Got it? Blight of apple and pear, Erwinia amylovora, Gall's agrobacterium species, soft rots, Erwinia aroideae, vascular wilts, Corynebacterium species, canker of citrus, Pseudomonas citri. So these are the various diseases which we see in plants. Got it? That is in mustard, cauliflower and all we see black colored dots. They are called black blight, bacterial blight. They are called black rot. Got it? The black spots turning of the cauliflower etc. into dark black color. Got it? So the second point you are going to learn is spoilage of food. The first one, diseases. Second one, spoilage of food. Food items exposed to air get contaminated and become unfit for human consumption. The main cause here is Salmonella species. Bacteria pass into meat from infected animals, mainly poultry, that is chicken, etc. and pigs. They affect the working of alimentary canal and cause diarrhea and vomiting within hours of consumption. That only we call it as food spoilage. Many bacteria spoil cooked food, milk, fruits and vegetables by the process of decay called fermentation, particularly in the summer, during summer. The spoilage may sometimes be to that extent causing food poisoning, namely botulism. The disease is botulism. Botulism is a very serious food poisoning due to a special bacterium sometimes found in tinned or sealed food. What is meant by tinned food? You might have seen Haldiram's Rasgulla. In a tin sealed container it will come. No, that is called tinned foods. Got it? The affected cans get distended at their ends with gas which gushes out on opening. The moment you open, the gas and the smell gushes out. Such suspected cans or tins of food should be completely discarded. Got it? That is why, children, preservation of food, got it, making the food free of disease causing germs is very, very essential. It is not only cooking, not only making, it is also very important to keep the food in a very good condition. Got it? Now, we are going for the next one. Decay of food. Decay of foodstuffs. That is spoilage of food. Yesterday's food, you will take the food in your food, that is lunch box. You will start eating. Sometimes you will forget to take it home. You leave it here in the classroom. Next day when you take it home, when you open, it will stink a lot. You will call that as spoiled food, stale food, isn't it? So that is called decay of food stuff. That is mainly caused due to saprophytic bacteria. Got it? Meat, food, vegetables and fruits all get decay. And another one cause called as biological weapons. They are called bioweapons. They are also called germ bombs. Got it? Some of the countries for the war, children, with the intention of the war, got it, they introduce biogerms into another country and cause diseases. Got it? Some of the diseases causing pathogens, their products are biological extract like certain proteins can be used to cause mass destruction of human populations, animals, or plants. Common pathogens which cause fatal means causing death. Fatal diseases can be picked up, multiplied in laboratories or industrial settings, and then can be used as bioweapons. Got it? Pathogen or other material chosen to make bioweapons are expected to have the following qualities. They should be easily obtainable. Everywhere they should be available. They should be highly toxic. The cost of their production should be low. They should be highly infectious and easily transferable from person to person. Weapon in form of an aerosol. Aerosol means the droplets found in the air. Their infection should be difficult to treat clinically with no available vaccine. Now you come to know which is the biological weapon or bioweapon. Yes, you are right. The COVID-19. Got it, children? Bacteria that can be used as biological weapon or bioweapon or Bacillus anthracis, Clostridium perfringens, Clostridium botulinum. Got it? Smallpox was like that only. It is introduced many years back when your grandparents were small. And it is later eradicated from the world. Got it? This viral diseases can be used as biological warfare. The smallpox virus may have been preserved in certain laboratories in the world from where it may be again obtained and again it can be spread. Got it? Ricin is highly toxic protein extracted from castor bean plant. It can be used as bioweapon. Got it, children? See how much is there about bioweapon. So now we are moving on to the next part of the lesson, that is how bacteria plays a very important role in agriculture. So far you have learnt the harmful effects. Now we are going to know about the useful part of the bacteria. The role played by the bacteria in the medicine. Got it? And in agriculture, all that. Is that clear? Useful bacteria. Got it? So first we'll learn role played in agriculture. First, nitrogen fixing bacteria. A special category of soil bacteria is found in living in small nodules on the roots of leguminous plants such as beans. In bean plants, etc. we find. These bacteria pick up free nitrogen from soil and atmosphere and convert it into soluble nitrates. These nitrates are used by the host plant and also by other plants sown later in the same soil. First they sow, first crop they take is leguminous plant and they leave the roots inside. You can see the picture left side, how nodules are there like pimples. So many nodules are there, no? They all fix the nitrogen. They are called nitrogen fixing bacteria. That is, they pick up free nitrogen from the air. You all learned 78% of air contains nitrogen. So that free nitrogen will be picked up and is stored in this nodules. First crop and the next crop also will be using this nitrogen. Is that clear? And certain nitrifying bacteria, free living bacteria in the soil like Azotobacter, Clostridium, got it? And they are called free nitrogen from the soil. First you have learnt the bacteria from the air. Now from the soil can also be converted into ammonia with the hydrogen, with the help of hydrogen, which in turn gets converted into amino acids and nitrates, enriching the soil, making the soil very, very fertile. So nitrogen fixing is done by two ways. One is by the free nitrogen from the air. Another one is free nitrogen from the soil. Is that clear? Now we are moving to nitrifying bacteria. Plants need nitrogen for synthesis of proteins, but they cannot make use of this free atmospheric nitrogen. So they obtain it only in the form of nitrates, which they absorb from the soil through roots. These nitrates are replenished in the soil. Just now we have learned recycling, replenished in the soil by bacteria through the process of nitrification. For this, some bacteria first convert nitrogen waste of animals and dead remains of plants and animals into ammonia. Ammonia is then converted into ammonium compounds by a type of bacteria called ammonifying bacteria. That is not given here. Ammonifying bacteria converts ammonia into ammonium compounds, which in turn are first converted into nitrates by bacteria such as nitrosomonas, and then nitrites and then nitrates. Got it? And by the bacteria such as nitrobacter. So here you are learning. So first into ammonium compounds by ammonification and ammonifying bacteria. Then nitrates. Nitrites is by nitrosomonas. Nitrates is by nitrobacter. Got it? And each of the step now you are going to have a look about with the help of nitrogen cycle. See, atmospheric nitrogen N2 first comes down as biological fixation by rhizobium, that is nitrogen fixing bacteria. That one way it comes into the soil by nitrates. Another way, plants will take these nitrates, they pass them in the form of proteins to animals. Both plants and animals' death occurs, die, and the decayed matter, that is by decomposes another set of bacteria. They convert them into ammonia, NH3. NH3 then converted into nitrites, then into nitrates. So two ways nitrates are coming into the soil. One is biological fixation by rhizobium. Another one is by the death of plants and animals. Plants take the nitrates, convert them into proteins. Proteins are consumed by animals. Animals will die and decay matter will first form into ammonia, ammonia into nitrites, nitrites into nitrates. The two ways how they are coming into the soil. And there is another way by industrial fixation. In the industries also, ammonium sulfate, ammonium chloride, like industries also they prepare and they use them as fertilizers. That's another one called an industrial fixation. So now how atmospheric nitrogen is entering soil we have learned. That completes only half of the cycle. Again the whole of nitrogen should go back now. That going back is done by another set of bacteria called denitrifying bacteria, which are present in the soil, which break down the soil nitrates to release nitrogen gas that enters into the atmosphere. Example is Pseudomonas. Got it? Pseudomonas is the bacteria which enter. Got it? Did you understand? Now, bacteria also play a very important role in industries, children. Got it? In industries, we are going to learn in medicine and industries. First, let us learn in industries. In industries, they mainly used for very extensively in industries. They are used as medicines, got it? And different flavors of tea, that is flavoring tea, cocoa tea, lime tea, ginger tea, you might have come across, no? That is called curing of tea, okay? And leather tanning, see, directly animal skin cannot be used as leather. That various steps they will clean, they change, that undergoes many steps, that is leather tanning. Leather tanning is primarily brought out by sunlight, but certain bacteria break down the soft perishable parts of the skin, that is hide, into a leather. Got it, children? And we are moving to medicines. In medicines, they are used as antibiotics. Antibiotic is a chemical substance produced by a living organism which can stop the growth of some disease producing bacteria and fungi. Got it? And it is also used as some of bacteria. Some of the bacteria they're also used as what we call serums and vaccines, that we are going to learn in the case of next part, that is in fungi. The definition of bacteria, serum, vaccines and all we are going to learn in the next part, children. Is it clear? Now we are going to learn a few techniques of food preservation. Got it? So you have learned now the role of bacteria in agriculture and in industries, whereas in the medicines we are going to learn in the next part. Is it clear? Now, so some of the preservation techniques now we are going to learn. So first technique is called sterilization. Boiling or heating at high temperature is called sterilization. Boiling water kills all bacteria except their spores. So higher temperatures, about 110 degrees at increased pressure, that is called autoclave. Usually we see this autoclave in clinics, hospitals, etc. Got it? And even in the pressure cooker, they kill even the spores. The canning industry employs heating to kill bacteria before packing and sealing the food. Got it? Second one is salting. Salting is a common method of preservation of foods like fish, pickles, cheese and so on for long periods of time. That is, you might have bought Amul butter, Amul cheese. It's highly salted. Lot of salt will be there, no children. Highly salted, like that. Dry fish. All these are the techniques. Salting is one of the techniques, children. By mixing these foods with salt, their biodegradation is prevented under normal conditions. That is why lemon pickle, mango pickle, lot of salt they will add, put it in the sun, isn't it? They dry them and then eat. So that is how they make pickles and all, preserved under the more salt. That is called salting. First one is heating to their boiling point, 110 degrees. Boiling and heating also kills the bacteria and preservation can be done. Second one is salting. Now third one, dehydration or drying. Most microbes cannot grow without water. Therefore, dehydration by drying of foods such as grains, meat, fish, vegetables is an effective method of preservation. Dried milk powder is another excellent example. So dehydration. First of all, the grains, children, dal, rice and all, you will see. They dry it and then pack it, isn't it? So dehydration is a drying. Drying in the hot sun is one of the best method of preservation. And milk powder is drying. It's a good example. Coffee powder also. Coffee bean will be dried and then made into powder. These are all cocoa powder. These are all best examples of cereals, grains. Best example for dehydration. Irradiation. A radioactive radiation has been tried in sterilizing certain foods without the food becoming radioactive. But the practice is not yet much used. In India, it is not used, children. But radioactive preservation is used in some of the foreign countries. Here, ultraviolet light is found useful and not only in sterilizing the air in schools, hospitals and factories, but also in killing molds, spores in bakeries. Nowadays we see, they say any COVID patient is there, they come sterilize. They leave some air, gas and all. You are seeing, no? Like that. Sterilization. By giving ultraviolet radiation also, sterilization can be done. Next method is pasteurization. Pasteurization is technique of partial sterilization applied usually to milk. Louis Pasteur found out, children, this method, that is alternate heating and cooling. Heat after few hours, cool it, again heat it, again cool. That is why it is called, because Louis Pasteur found this, it is called pasteurization. The milk is heated to a temperature of about 60 degrees C for a period of 30 minutes and then chilled quickly and then stored. Pasteurization kills a majority of bacteria that are present, including the disease-causing ones. Pasteurization does not completely sterilize milk. Soaring bacteria may still be present in it. By keeping the milk sufficiently cool, they do not multiply. That is why the packets, those which are coming, packet milk, is pasteurized. Even then, we keep them in the refrigerator because pasteurization completely will not kill the bacteria. Refrigeration, keeping it in a cold temperature. Microbes or bacteria or molds means fungi, do not grow and multiply at or below the freezing point of water. Chilling of milk and keeping vegetables and fruits in cold storage are popular methods of preserving them. You might think 2 packets, 3 packets in our house we'll keep. What they do for a lot of vegetables means the whole house will be converted into cold storage unit, children. Got it? So in such places they keep the vegetables. Frozen fish, frozen vegetables keep the fresh for long time. So you might have seen in metro and all, the whole unit will be frozen. Domestic refrigerator cools up to 0 to 5 degrees C for short-term storage. Deep freezing cools at minus 20 degrees C to minus 30 degrees C for long-term storage. Got it? The ice creams and all, they use this long-term storage. Domestic refrigerators means the refrigerators which we find at home in our houses. Next method is strong concentration and chemicals. You might have seen this, what is that, rasgulla, jangri, jalebi and all are put in a strong concentration, sugar solutions. Jams and pickles do not get spoiled because they are using strong concentration of sugar and salt respectively. The increased concentration of the solutes causes plasmolysis and death of any bacteria or mold that may creep in. Plasmolysis means the water comes out and the mold become dry and they die. Got it? Sometimes preservatives like sodium benzoate are also added to the tin foods and squashes. Got it? Thank you, children. So next lesson we are going to learn is harmful and useful effects of fungi, that is economic importance of fungi. Is it clear? Go through this lesson, read thoroughly. Sometimes some places page numbers are given. Carefully you go through. Any doubts you can ask in your doubt session. Thank you.